Hello members, welcome to our discussion of today. We're going to proceed with our discussion on the nucleic acids from where we stopped on the discussion of nucleotides. So we're going to proceed with a look at the polymerization of nucleotides, how nucleotides are joined together from one to another to form the long strand of nucleotides, which shall be now called as the nucleic acids. Now, remember we discussed that a nucleotide will be consisting of three things, namely the nitrogenous base, the pentose sugar, and the phosphate group. And each nucleotide will have either one or two or three phosphate groups, as we saw in our last discussion. So, how will these nucleotides be joined together to form the long strand of nucleotides like this, which we shall then subsequently call as the nucleic acid? The polymerization here will be occurring through formation of what we call as the phosphodiester bond. Phosphodiester bond is the bond that joins together any two successive nucleotides in the polymer of nucleic acids. Now, to join this nucleotide to this other nucleotide, you will need a phosphodiester bond between them, isn't it? And also, to join this nucleotide to this other nucleotide here, you'll also need a phosphodiester bond. You'll also need a phosphodiester bond. And also, in this other case, to join this nucleotide to this other last nucleotide here, you require a phosphodiester bond being formed between them. Now, how does this phosphodiester bond form? The phosphodiester bond formation requires energy to facilitate it, and that energy to facilitate that phosphodiester bond formation is obtained from from hydrolysis of what we call as phosphoanhydride bonds. In fact, remember we discussed about phosphoanhydride bonds as the bonds that join the second phosphate onto the first phosphate in the nucleotide, and then also the third phosphate onto the second phosphate in the nucleotide. So those phosphoanhydride bonds, when they are being cleaved off, they release sufficient amount of energy that will then subsequently facilitate the formation of this phosphodiester bond. Now, after these phosphodiester bonds have been formed between successive nucleotides, remember the nucleic acid will not have only 4 nucleotides as shown here. They will have thousands and thousands and thousands of nucleotides joined together, but still the bond that joins together, the bond that joins them together, it is still the same. The phosphodiester bond is the same bond that joins all nucleotides in the nucleic acid. Now, once the nucleotides have been joined like this, what will happen is that they will then form a strand of nucleic acid, which we shall then call as nucleic acid. And in the case of DNA, 2 strands shall be intertwined together to form the DNA molecule, isn't it? 2 strands shall be intertwined together to form the DNA molecule. But for the case of RNA, it shall only be consisting of 1 strand of nucleotides joined together. So whether it's 1 strand in the case of the RNA, or even if it's 2 strands in the case of DNA, these 2 molecules, RNA and DNA, would be considered as the nucleic acids, which we have seen previously as the 2 types of nucleic acids which exist within our cells. So now, after the DNA has been formed like this through many nucleotides being joined together, and then the DNA strands intertwining among themselves or between each other to form the structure as shown here, then this DNA shall be required to be packaged where? To be packaged within the cell, and specifically within that small organelle which we call as the nucleus. Remember, the nucleus is present in every eukaryotic cell. So in that nucleus, we'll be having a lot of DNA molecules inside it, and they occur there in the form of chromosomes. So chromosome formation requires that the DNA interacts with specific proteins which are going to help in packaging that DNA into smaller structures which we call as the chromosomes. So the chromosomes are then subsequently arranged together within the nucleus to form the genetic composition or the genetic makeup of the cell, especially the eukaryotic cell. So those proteins which we are calling as histones, which are involved in the packaging of DNA, if they are not present within the eukaryotic cell, automatically you know that that eukaryotic cell will not be able to package its DNA into a small or short molecule which is in the form of the chromosomes, and therefore that will be posing as a size constraint to the eukaryotic cell because of the fact that the cell is a very small a very small structure which requires that everything within it be miniaturized or be made small in order to fit within that cell structure. Now, after the DNA has been arranged in form of chromosomes and packaged into the form of chromosomes. Now, what happens subsequently? This DNA will need to be formed into the subsequent structures when time requires. Especially, that form is to convert the DNA into messenger RNA and then subsequently convert that messenger RNA into proteins through what we call as gene expression. So, this gene expression process, we shall see it as the stepwise process by which DNA is converted into messenger RNA, and then that messenger RNA, through the help of the ribosomes, will be translated into the proteins. So, the process through which DNA is converted into messenger RNA and then messenger RNA converted into proteins is what we call as gene expression, and that stepwise process is also called as what we refer to as the central dogma. Now, this process of gene expression, remember, it involves two steps or two processes. The conversion of DNA into RNA, which we specifically call as transcription, and then the conversion of the messenger RNA into proteins, which we call as translation. So, combined together, transcription and translation help to convert a DNA molecule into a protein molecule which is functional in nature. It could be the protein present in your hair, it could be the protein called as amylase, maybe present in your saliva, it could be any other protein. Even the keratin which is present on your nails, on your fingernails, is basically made through this stepwise process of converting DNA into messenger RNA and then into that keratin, which is the protein resulting there. Now, there are some cases whereby you will see that the process of transcription and translation is interfered with. So once that process is interfered with, usually it will result in no formation of the required protein, and that has sometimes detrimental effects on the individual. For example, if you consider the person suffering from lactose intolerance. In lactose intolerance individuals, usually the problem is that they cannot form a certain protein which is called as lactase. In that case, it's an enzyme. They cannot form that lactase enzyme which is required for the metabolism of lactose, and therefore they suffer from lactose intolerance. Their inability to form the lactase enzyme in that case of lactose intolerance will be usually due to a distortion in the structure of their DNA in the form of what we call as a mutation. So the mutation results in a different type of messenger RNA, or a messenger RNA that has a slightly differing nucleotide sequence, and therefore that nucleotide sequence, when it is translated, it results into the formation of a defective protein, a total defective protein, not the desired lactase which is required for those individuals to actually metabolize lactose in that case. And you know the consequences of the lactase absence in the people suffering from lactose intolerance. You are aware that they experience hard life, especially when they consume lactose, especially milk. If you give a child milk and that child is lactose intolerant, usually you see them vomiting and having diarrhea also. Even in adults, those adults who have lactose intolerance, they usually have the same features. They usually get vomiting and even diarrhea when they consume milk products because of their inability to metabolize that lactose due to the absence of the lactase enzyme. So at least now you have a quick understanding of how the nucleotides are polymerized to form the DNA, of which case that DNA will then be expressed to form the proteins, of which those proteins will have specific functions within our cells. If those proteins are not formed, for example, in the case of lactase, then there will be consequences in the form of inability to perform the function that that protein was supposed to be able to help you perform under normal circumstances. So from there, from the video which we saw about gene expression, we shall also understand how that process of expression requires the messenger RNAs, the tRNAs, and the ribosomes to proceed normally. Thank you. We shall proceed from there.